Thank you, Bakos. Santosh Kesri. I'm actually joined as a consultant to help the company. I actually know of his product for about 20 years because patients have used it since my day of it is. So this is a very different product than what we're talking about at this conference. This is a natural product rather than a defined chemical entity. And I think we know the history of oncology is such that many of the old chemotherapy drugs and even some of the targeted drugs that we use are actually derived from nature, whether it's from plants, fungus, bacteria, antibiotics, etc. So it's not unusual that natural products could have positive effects in humans, on the microbiome, on the cancer directly, on the immune system. And that's really what this product is about. Having had patients experience positive effects and the anecdotal information about patients responding, I think this company is really trying to get it into the clinic as quickly as possible in formal clinical trials. And I'll leave it up to Dr. Magnifico to explain the history of this approach, and then also to then show phenotypic preclinical data and the clinical thought and the executive.
Santosh, thank you very much. Good early afternoon to everyone. First of all, let me offer our thanks to your organization for this symposium. Well, as to showcase our product and our efforts, my name is Michael Magnifico. I'm one of the medical advisors for Pacific Marine Biotech, located in Northwest Connecticut and also in Australia. So PMB, as we would like to call it, is a literal description of our product. We call it Immune 12. It's a complex marine product that's freshly sourced from the waters off Vanuatu, which is west of Fiji. And it's composed of two sea cucumbers, sea urchin, sea squirt, garnishy seagrass. It's chosen and distributed from there. The basis of this product is traditional Chinese medicine, and its use has cured the stomach cancer of our founder, Mr. Samuel Brand. Yes, I said that. He and a father started this company producing and marketing this product as a nutraceutical, a dietary supplement in 1881. In more official designations, in 1991, it was granted an OST-ARD number, which is equivalent to a level of an FDA approval as an arthritis drug in Australia after successful clinical trials there. In 2012, it was granted orphan drug status by our FDA in smoldering myeloma after a review of successful clinical trials in New York at St. Vincent's, Mount Sinai, and NYU medical centers. It should be noted that no toxicities were noted in any of these clinical trials. And further preclinical work at Charles River, Nova University at Florida, and also in UNC, which you will hear about shortly, have demonstrated no toxicity. This product has been commercially available. It is drinkable. It's been used on a voluntary basis for over 40 years. Multiple clinical conditions have been treated with it. People take it in a voluntary fashion, but it has been noted that unusually good results with certain tumors, including the glial tumors, GBM, and DIPG have been noted, and that provides the impetus of the clinical work that you will see showcased here. Following me, Professor Andrew Satterlee of UNC will describe ongoing clinical studies there, preclinical studies, I should say, to elucidate the composition and the biological interactions of this complex product. Next, Dr. Janet Schloss, our Australian colleague, will present her experience with Immune 12 and describe the clinical trials now opened illustrated phase 2. Finally, Mr. Samuel Grant, who is the founder of this company, will summarize the philosophies and goals. Thank you very much.
Interestingly, although this has been a commercially available product as a supplement for decades, there's not been very much preclinical data generated on how the product works, what's in it, what the mechanism of action is. And so my lab, my core facility at UNC, have started just recently doing some work alongside PMB to figure out some of this. We want to know, what's the anti-tumor potency of immune 12 like? Can we characterize the complex makeup of this compound? And then figure out how it's working. What's the mechanism of action? Are there several? There's so much that we don't know, even though it's been used and we're seeing clinical benefit in it. There is a little bit of preclinical data that's already been published. This is a paper from 2019 that shows an orthotopic model of prostate cancer can be suppressed by oral gavage of immune 12, and that it affects the protein profiles of cancer cells as well. There's a couple other papers that have been out in the last few years, one on the myeloma that Dr. Magnifico just alluded to, and another case study and review paper from last year by Dr. Sanihagi. So in our lab, we started from the very beginning. Can we put some of this immune 12 on cells in vitro and see killing? Yeah, we can. Fairly potently and in a variable manner where different cell types have different potencies, different sensitivities to this drug. In 5 different GBM cell lines, in 2 ovarian cancer lines, in a breast cancer line, in lung cancer, and in pancreatic cancer, we see over and over again that 1 to 3% or less of immune 12 does have some direct cytotoxicity on these cells. And the difference in the sensitivities offers us an opportunity to try to figure out what's going on here. Do the most sensitive cell lines react to immune 12 in a different way, a fundamentally different manner than these more resistant cell lines that still are killed at a fairly high rate at a low percentage in vitro? We've just started this. We've started doing some Western blots to look at protein upregulation profiles and downregulation profiles. We think it may partially function via PJUN, PP38 induction, as known to show apoptosis in some cell lines. But we're hot on the trail of what the mechanism is, and we're starting some global kinase studies. Because right now we're just kind of shining a flashlight in it in a very dark room. We want to figure out what's going on, and we've got some experiments lined up to do that. We've done a 28-day treatment study in mice, in healthy mice, immunocompromised and immunocompetent mice, given immune 12 by oral gavage every day. But we're not seeing any toxicity by BioWeight or a lot of other metrics, which is really encouraging and supports the data that we're getting in people as well. We've just started some in vivo orthotopic models of brain cancer and other cancers, and we're seeing this is approaching significance here is these 2 untreated and treated arms of this study. But we're giving immune 12 orally to these mice, and we have a tumor implanted orthotopically in the brain, and we're seeing some tumor growth inhibition. This is really encouraging. It's the first time we've tried this. Got many other studies lined up. And we also know that immune 12 contains a multitude of proteins. We've done a lot of proteomic analysis and metabolomic analysis, and we've even identified several instances of known tumor suppressors within immune 12. We don't know what the active component is or which of many components may be active, but we're hot on the trail of this as well. There is a lot of future directions. My work is just beginning. The work of my university, my lab, and the lab tool we're collaborating with is just beginning. We've got a dozen or so people working on this across four facilities with 6 departments at UNC, looking not just at GBM, but focusing on GBM, looking at batch-to-batch consistency, working with the manufacturer, doing a lot of in vivo experiments. Right now, we've got brain, ovarian, breast, and pancreas models all getting ready to begin. We're looking at proteomic characterization and even the microbiome that may be affected. You don't know how this product works, so it may work indirectly through other mechanisms that are just direct cytotoxicity. And then MILAX, bread and butter, is our technology to test new drugs on uncultured patient tumor tissue we get right from the hospital. So we're going to start doing work with those as well. So I'm willing to take questions afterward, but I'll let this get up to Janet now.
So I'm going to introduce our clinical component, and today I'm going to talk about some case studies and our phase 2 trial, which we've got up and running in Australia. Before we do that, I want to sort of introduce us to how I came about doing the phase 2 trial. I've known about this product now for about 8 years. I have prescribed to some of my patients in regards to the literature and blood collection we're studying for. I have reached out, or a colleague that reached out to me from Canada and said this company is actually looking at someone who's ready to run a trial under the cucumber but for glioblastoma. And I thought that it might be just been doing it, but I said, sure, as she talked to the company. So they come in, they actually show me the case studies, what the information was, and I was actually very interested. So she took it on. From that position, I was actually able to get some of the free product for some of my patients. So I had 8 patients who on case studies, which I'll actually talk about today. And we've actually started looking and started putting together the phase 2 trial. In doing so, in that meantime, they actually published the case studies that were shown to me, as well as the literature which you can actually find in the Oncogem. So of the 8 patients who had consented to be able to use their information or case studies leading up to our clinical trial, I'm going to present to you 2 case studies today. These are 2 that I first put on to the product. One is a recurrent GBM, which is case study 1, and then other one that follows is a newly diagnosed glioblastoma patient who follows our group 1 for our clinical trial. As you can see, the reason I've actually picked the first one is because the procedures actually have quite a remarkable outcome, which you see when I present the case. She's 42, methylated. She had completed first-line treatment. Before she completed her 12 months of temozolomide, she actually had recurrence. She went on to the TAC1 trial. After 1 month of being on that trial, she had a complete new butterfly tumor commence and was quite extent at that stage. She commenced on the Mu12 on the 5th of December. Case study 2 is a male. He was newly diagnosed. He had 3 tumors. From there, he only had one of the main, which was actually debulked. He's hypermammillated. He has EGFR, which you've got a lot of this component. And he started just before he commenced high-dose temozolomide, post-MRI from radiation and reverse temozolomide. So looking at the first case, she was diagnosed in June 2022. As I said, she had the craniotomy. She went through first-line treatment. Just before that finished in July, they saw potential regrowth. By August, it was confirmed. She was then taken off temozolomide and was eligible to go onto the TAC1 trial, which she started in October. In November, 1 month after she started, they saw extensive growth, new tumor in the corpus worksome. They stopped the trial, and she was actually went down to Adelaide for the actual trial itself. And home in Brisbane, her medical oncologist at that stage had reached out to the company to see if she can get compassion care for some immunotherapies. As she was deteriorating quite quickly, she ended up having 2 shunts having to be put in, and then we started the Mu12 on December. On December 11th, she started Ipi-Novo. The 29th of December, obviously there was continual growth that we saw at that stage. I saw her in early January. I actually did think that she was going to live. As you can imagine, you've seen all these type of patients. She was not being able to move. She had dysphagia, incontinence, fecal and urinary, and I thought she had battle big to go. In mid-January, her wife noticed some noise in the room through the night and woke up to see this particular patient standing and going towards the door. She said, "What are you doing?" She said, "I'm going to the toilet." There'd been a light switch, and from there she actually progressed quite rapidly into coming back into normality. The February MRI actually showed reduction in disease, and after her 4 lots of Novo, by the way, she continued with immune 12 all the way through this, she was granted a maintenance dose of Nevo up until January 2025. So what's remarkable about this case is that in the main last year, her MRI showed no evidence of disease. And that has actually continued out into her last MRI, which she got just before she came over the year. She's actually Mexican. She really wanted to come back to Mexico. Over the Christmas period this last year, she spent a month in Mexico, still continuing to take the immune 12. She still continues to take that today. Right now, she's living her best life. She's back working and living normally. I've never seen a case like that yet. These are her MRIs. You can actually see the first one, which is her initial one, the 9-centimeter tumor in myeloma. And the 7th of November is closely type 1 trial, where you can actually see the new growth of the butterfly tumor. The 29th, again, continued growth. The February one, you can start to see that it's reducing. The May, August, and November show no evidence of disease. Happy to show you, got all the MRIs. Case study 2. So this particular case study, like you said, was a newly diagnosed male. He was in the Air Force, and he was diagnosed with 3 tumors. They evolved the first one, it's just the biggest one. The other 2 were left for treatment. He had his MRI post-radiation low-dose temozolomide and commenced that immune 12 day after his MRI. The first MRI afterwards, he wasn't able to get contrast. We didn't get a good response at that stage.